All right, so we're going to wrap up now our section on DNA structure and replication by talking about mistakes that are made and sometimes how those mistakes are fixed by ourselves once the mistakes are made. So, the first thing to realize here is that because of how DNA replication works in that semi conservative manner, there really is not a lot of potential for mistakes to be made in the first place. There really aren't that many mistakes or mutations that occur during DNA replication at all, really. But when the mistakes do occur, and they do occur from time to time, we think about once out of every 10 million or so attempts, those mistakes, far more often than not, are repaired. They're fixed by our cells, so they don't actually have a chance to be expressed as part of our overall physical appearance. So, one of the reasons why DNA can be fixed is because the enzyme that builds the DNA polymerase, is actually going to have several different kinds of repair mechanisms. DNA polymerase is able to proofread and find places where mistakes have been made. So if, for example, you got a cytosine put in where there should have been an adenine, DNA polymerase can recognize that and fill in the correct base where it's supposed to be. So it doesn't actually have a chance to show up as part of the physical appearance or behavior or whatever the trait might be. So there are certain kinds of things which can increase the rate of mutations. We call these things mutagens. They can be things like radiation, X-rays, gamma rays, UV rays are all mutagens. Various types of chemicals can be mutagenic as well. And really all that means is that these mutagens are able to somewhat increase the background rate of mutation. So instead of having a mutation that occurs 1 out of every 10 million or so attempts at copying DNA, maybe a mistake happens once out of every 10 once out of every million or so attempts at copying DNA. It goes from 1 out of every 10 million to 1 out of every million. And that's a 10 times greater increase in the rate of mutation, which is pretty substantial. There are other ways to repair DNA. There are other proofreading mechanisms, repair mechanisms, and so forth. We'll talk about one of them just really, really quickly at the end of this section, but there are many, many different ways to repair DNA. And all of that really, in the big picture, sort of lets us realize how important this DNA stuff really is. We wouldn't, expect natural selection to favor lots and lots of repair mechanisms if DNA was some kind of arbitrary molecule that didn't really matter all that much. The reason why we have so many repair mechanisms is because DNA is such an important molecule. Now, as far as repair mechanisms, there are two main types. There is specific repair and there is non-specific repair, right? So specific repair mechanisms are those that target very specific kinds of damage. So, for example, we have in some cases what can be described as being a thymine dimer, right? So thymine is one of the bases that makes up DNA. It's the T of the ACGTNA. And dimers are situations where two things are linked together to create one larger thing. So a thymine dimer describes a situation that occurs when two thymines that are next to each other in DNA actually bond to each other as opposed to bonding to adenines on the complementary strand. And the thing that causes thymine dimers to occur is UV radiation. So thymine dimers are very common that occurring in the skin, but not really very common anywhere else in the body because the UV radiation only has enough energy to penetrate the outer layers of our skin. So therefore we see this particular type of damage occurring in the outer layers of the skin. Now, there actually is a repair mechanism here that is set up to fix thymine dimers. It involves an enzyme called photolyase. So photolyase is Photolyase, photolyase. Photolyase has the ability to break a thymine dimer and therefore force those two thymines to go back to bonding to adenine on the other side on the complementary strand. So that's one example of a thymine dimer specific repair mechanism. What's kind of interesting about this is that the photolyase has to have an energy input in order to break the thymine dimer, and the energy that it uses is actually sunlight. So sunlight not only causes this particular kind of damage, the thymine dimer, but it also powers the repair enzyme, photolyase, which is used to fix the thymine dimer. We also have several what are also called non-specific or just generalized repair mechanisms where it's just sort of like a generalized proofreading process that enzymes can use to fix any sorts of damage that might occur in a cell. So a lot of times if you have a base bonding to the wrong base on a complementary strand, the DNA molecule will take on an abnormal shape, and various types of proofreading enzymes will notice that and repair it.